Morning, everybody. I'm going to take a class on microbial growth and effect of environment on microbial growth. This is included in undergraduate microbiology courses, semester 4. So we'll discuss the elemental macromolecular compositions of a bacterial cells. Here we can see periodic tables. We know what is periodic table, but here if we can see different element are color coded. So and this is the significance of the color code is those elements are required for microbial growth, and you can say it is a chemical makeup of a cells. We know that carbon, nitrogen, and other elements, like here, the yellow coded elements are essential for all microorganisms like hydrogen, carbon, nitrogen, phosphorus, sulfur, oxygen. And here use different color codes like yellow, like light pink, light green, like light blue, and all are coded all are coded color is indicate that whether it is essential or whether it is unessential or whether it is not. So so if you see the next slide is here, here it is presented macromolecular composition of a cells. What is the composition of a macromolecular, or what is the compositions of a cells? The cells, there are macromolecules like protein, there are macromolecules like lipid, there are macromolecules like polysaccharide, lipopolysaccharide, DNA, RNA are present inside the cells, and they are present in presence of different dry weight, and this is the relative abundance of macromolecules in a bacterial cells. This data actually from Escherichia coli and Salmonella typhimurium. And what is the elemental composition of E. coli cells? You can see this is the cartoon diagram where 50% carbon, 20% oxygen, 14% nitrogen, 18% hydrogen, 3% phosphorus, and so on and so forth. So, and this is the distributions of elements in an average E. coli cells, and that is reflecting their dry weight. So here we can say that the role of and the composition of equally bacterial cells, equally bacterial cells of element of elements. So now we will try to learn the micronutrients needed by microorganisms. What are the micronutrients like trace elements and growth factors needed and their functions? We will discuss one by one the name of microelement and growth factor and their functions. So such as boron. Boron is a trace element. It's auto-inducer. What is the function of boron? It is an auto-inducer for quorum sensing in bacteria. It is also found in some polyketide antibiotics. Cobalt. Cobalt is actually vitamin B12 in function in vitamin B12 trans-carboxylase only in propionic acid bacteria. Copper, Cu. What is the role of Cu? Copper in respirations, cytochrome C oxidase, in photosynthesis, plastocyanin, and some superoxide dismutases. Next, iron. What is the role of iron? Iron function in cytochromes, catalases, peroxidases, iron sulfur proteins, oxygenases, and all nitrogenases. Next, what is the role of manganese? Manganese is the activator of many enzymes, components of certain superoxide dismutase and the waste splitting enzyme in oxygenic phototrophs like photosystem 2. We know photosystem 1, photosystem 2. Next one, molybdenum. What is the role of molybdenum? Molybdenum having a certain flavin containing enzymes, some in nitrogenase, nitrate reductase, sulfide oxidase, and DMSO, TMAO reductase, and some formate dehydrogenases. Next, the trace element is nickel. What is the role of nickel, or what is the function of nickel? Most hydrogenases coenzymes F430 of methanogens, carbon monoxide dehydrogenases ureases. Selenium, selenium Se. What is the function of selenium? Role in formate dehydrogenases, some hydrogenases, the amino acid selenocysteine. Tungsten, some role. What is the role of tungsten? Some formate dehydrogenases, oxo transferases of hyper thermophiles. Vanadium, vanadium nitrogenases and bromoperoxidase. And zinc, what is the role of zinc? The carbonic, it is a function in carbonic anhydrase, nucleic acid polymerase, and many DNA binding proteins. This is the total scenario of the trace element and their functions. Now, what is the role of growth factor? Now, what are the different growth factors? What is the different growth factors is actually important for bacteria. First one is PABA. PABA is para-amino benzoic acid, folic acid. So what is the role of PABA? It is the precursor of folic acid. Here we can see there are a lot of antibiotics used nowadays, and most of the antibiotics are actually PABA inhibitors. So that means PABA is para-amino benzoic acid is very important, or folic acid, the very important precursor of folic acids, and one for bacterial growth. And folic acid, one carbon metabolism, methyl transferase. Biotin, what is the role of biotin? Fatty acid biosynthesis and some CO2 oxidation reactions. B12, cobalamin. What is the role of cobalamin in bacteria? One carbon metabolisms and synthesis of deoxyribose. And what is the role of B1? B1 is actually called thymine. B6 actually called pyridoxine. What is the role of B1? It is function in decarboxylation reactions, whether B6 in amino acid or keto acid transformations. Next, the growth factor is nicotinic acid, whether it is called niacin, is a precursor of NAD+. The next is riboflavin. Riboflavin is the precursor of FMN or FAD. Pantothenic acid, precursor of coenzyme A. Lipoic acid is a decarboxylation of pyruvate and alpha ketoglutarate. Vitamin K is its role in electron transport. We know electron transport is very important for energy generation, like ATP generations. And then coenzymes M and B role in methanogenesis. F420 and F430 is a growth factor. This also role in methanogenesis. So this is the overall scenario of different elements and different growth factor and their role in microorganism. So from previous two slides, we have learned that what are the elements are important for microorganism and their role in their growth, and also we have learned the different trace element, not only the major element, the minor element or trace element, and also the growth factor having different functions in bacteria. So in bacteria grow in naturally in the nature, but for our research purpose, for our study purpose, we grow bacteria in our laboratory. So how we grow in our the bacteria in our laboratory, we need to know how to grow them. So for these reasons, we will learn some few slides about their growth. How can we maintain about their growth in the laboratory? We need to know the few things like what is the culture media. We used to see, we used to tell this word culture media very often. But what is culture media? The media is a source of nutrients to support the growth of the microorganism in vitro. The media helps in the growth and counting of microbial cells, selecting of microorganism, and survival of microorganisms. The culture media can be liquid or gel. So is this clear? So what is culture media? The media is a source of nutrient to support the growth of microorganism in vitro. We use the two words in vitro and in vivo. So in vivo means inside the cells, and in vitro means outside the cells. But here we are growing cultures, we are growing bacteria in a laboratory condition. So you can say it's a in vitro. And the media helps in growth and counting the microbial cells, selecting of the microorganisms, and survival of the microorganisms. The culture media may be liquid or may be gels. So now we need to understand what are the common ingredients of culture media. So there, what are the ingredients? It consists of culture media often consists of peptone, beef extract, yeast extract, distilled water, agar. So here we will learn one by one what is the role of these compositions. Peptone is why peptone is used in culture media. It is the source of carbon and nitrogen. Beef extract. It is the source of amino acid, vitamins, and minerals. What is the role of yeast extract? Source of vitamins, carbon, nitrogen. Need distilled water, and also agar. Agar is the solidifying agents. So this is the common ingredients of culture media. So here we can see the slide of classification of culture media. What are the culture media has been used, and their classifications. And these classifications based on the classification is based on here consistency, based on ingredients, and based on O2 requirement, oxygen requirement. Okay. And we will learn that special medium, what are the special medium also used, and it is depend on ingredients. So again, I am repeating that the classification of culture media. Here we are see that different there are different culture media, but the classification is based on consistency. Number two is ingredients. Number three is O2 requirement, and also the special media in based in ingredients also classified. So in consistency, we can classify it into solid medium, liquid medium, semi-solid medium. In ingredients, it is classified into simple or basal medium, complex medium, synthetic medium. And on classification of based on O2 requirement, aerobic medium, anaerobic medium. So we will learn details one by one in every medium, and they are example too for the learning purpose. Next, special medium can be divided into enrichment media, selective media, indicator media, differential media, and transport media. So, but we learned the classification of different culture media, right? But in the laboratory conditions, we need to prepare the culture media. So, how we can prepare the culture media? How we can prepare the culture media? Here is the different steps which required for preparation culture media. What is the number one step? Weigh the amount of ingredients powder on weighing machine in the laboratory. Number two, then dissolve the ingredients in distilled water. After dissolving the ingredients in distilled water, then you need to adjust the pH of the medium if needed. If you needed to adjust the pH, like some organisms grow in pH 3, some organism can grow in pH 4, can 5, 6, 7, 8. So different pH requirement from them. So in that, for that purpose, we need to adjust the pH of the medium, because if you don't adjust the pH, so then bacterial growth will not be possible. So pH is an important factor for bacterial growth. Next, you need to add agar and boil to dissolve. You need to agar to boil for dissolve, and we have already learned what is the role of agar in the previous slide. It is a solidifying agent. Next, pour the media into flask. pour the media into flux. Next, autoclave the media when fully dissolved. Ingredients fully dissolved. Next, sterilization is required. Sterilization is required because if you don't sterilize through autoclave, the unwanted microorganisms will be grow that are previously present in the media during the preparation. So sterilization is done in autoclave to prevent from contaminations at 121 degrees Celsius for 15 lV pressures. In 121 degrees Celsius, 15 minutes at 15 lV pressures. This is the principles of autoclave. And after the autoclave, place the culture flux in laminar airflow. So laminar airflow is a place where we can use for culturing the media to grow the to aseptically transfer the media. Okay. Next, sterilize the laminar airflow with 70%. Sterilize the laminar airflow or disk with 70% alcohol. The 70% alcohol is here used as disinfectant agent. Next, a bit cool down the media and pour into sterile petri plates for solidifications. So then sample is ready to spread or streak. Now you need to use inoculation loop on the medium for identifications or isolation of microbes. And then after using inoculations loop for inoculation the media, you need to shield the petri plates with paraffin and label them and keep them inverted at incubator for 37 degrees Celsius for 24 hours. And next day we need to observe the result that the colonies formation is visible on the medium or not. And this is the overall procedure. This is the overall procedure how to prepare culture media and how can you inoculate a culture media and grow them on them and expecting a colonies from the culture media. Now we will learn one by one the different culture media as we have discussed in the classification slide. Now, what is defined medium? A defined medium has a known quantity of all ingredients like carbon source, glucose or glycerol, and nitrogen source, ammonium salt or nitrate as an inorganic salt, and the medium needs in metabolic, nutritional, and physiological growth experiment. So this is called defined media. I am repeating this one. A defined medium has a known quantity of all ingredients. Known quantity of all ingredients, everything is known, like carbon source, as for example, glucose and glycerol, the nitrogen source, as for example, ammonium salt or nitrate as an inorganic nitrogen. The medium needs in metabolic, nutritional, and physiological growth experiments. Example of a defined media, what is the example? Japet dogs medium. Next, what is an undefined medium? So we have learned the defined medium is previously. Now, what is undefined medium? The medium has different complex ingredients is unknown quantities. For example, yeast extract, beef extract, various salt and enzymatic protein. For example, potato dextrose agar, macron ki agar. These are all ingredients are undefined. So is it clear? So what is the defined medium and what is the undefined medium? Defined medium has a known quantity of all ingredients like carbon source, like nitrogen source, and in undefined media, the medium is different complex ingredients is unknown quantities. For example, yeast extract, if you use yeast extract, the quantities will be unknown, the ingredients will be unknown. If you use beef extract, the quantities will be unknown. And if you use various salt and enzymatic proteins, the potato dextrose agar, macron ki agar, it is macron ki agar. So as for example, for undefined medium. Now, what is the complex media? This media is other than basal media and it has added ingredients to bring the characteristics of microorganism with unique nutrients. With unique nutrients. So what is the complex media? This media is other than basal media, and it has added ingredients to bring the characteristics of microorganism with unique nutrients. So you can drop down a comparison between defined media, between undefined media and complex media. Now, what are the types of culture media based on we will learn now the types of culture media based on consistency or physical state looking like solid. If it is looking like solid, it will be solid media. If it is looking like semi-solid, it will be semi-solid media. And if it will be looking like liquid, it will be liquid media. So solid media, now what is the solid media? What is the principle of solid media? The solid media, it is used for the isolation of bacteria as a pure culture on a solid medium. Robert Cox realized the use of solid media very fast. Here, agar is used to hardening the media, and we will use 1.5 to 2% concentrations. Solid media allow the growth of bacteria as colonies by sticking on them, and it is solidified at 37 degrees Celsius. Agar is an unbranched polysaccharide. What is agar? Now we need to know the agar. So what is solid media? It is the isolation of bacteria as a pure culture on solid medium. But how can we make it solid? So how can make it solid? We can use agar for its solidification. So now we need to learn what is agar. Agar is an unbranched polysaccharide extracted from red algae species like Gelidium. Colonies identification is done on this, and colonies identification can be done in this media. And Robert Koch realized the use of solid media. Now, what are the example of solid media? Like nutrient agar, like McConkey agar, like blood agar, like chocolate agar are the example of solid media. And the growth of bacteria on solid media appears very smooth, rough, mucoid, round, irregular, filamentous, and punctiforms. So these are the different types of growth on the solid media we can observe when we can try to grow the culture on solid media. And now, what is semi-solid media? What is the principle of semi-solid media? This media shows the motility of bacteria and cultivation of microaerophilic bacteria. The media shows the motility of bacteria and cultivation of microaerophilic bacteria. This media has an agar concentration of 0.5% or less, and it is a jelly consistency. So try to find the difference. In solid media, the agar percentage is higher compared to semi-solid, like 1.5% to 2% we are used for mostly. But yes, in semi-solid media, only the 5.5% or less. That means it will be not so solid as compared to solid media, and it will be like a jelly-like consistency. And this why this is has been used. This media has been used for grow the bacteria which have a motility. We are looking for motility, so the media will show the motility of bacteria and the cultivations of microaerophilic bacteria. So what is the now? What is the example of semi-solid media? The stewards and amines media, hungs and Leifson's oxidation fermentation media, mannitol motility agar, these are the example of semi-solid media. And these are the growth of bacteria in semi-solid appears as thick lines in the medium. We will not find colonies as semi-solid media. Here we can found a thick line in the medium when microorganisms will grow. Now, what is the role of liquid medium? The principle of liquid media, this media shows the growth of a large number of bacteria. It is called, this bacteria is used for growth of large number of bacteria and mostly used in the laboratory is liquid media. And it is called broths, and that allow the bacteria to grow uniformly with turbidity. And the growth occurs at 37 degrees Celsius in an incubator for 24 hours. And liquid media does not have the additions of agar. It is the fermentations studies. So you can, if you compare with solid media and liquid media, here no agar has been used, and most of the bacteria grow in the liquid media, and this media is actually called broth, and you can grow the bacteria uniformly, and you will get the turbidity, uniform turbidity from liquid media, and the culture can grow in various temperature. The growth optimum temperature is 37, but you can grow in various temperature. Now, what is the example of liquid media? The nutrient bottle, widely used. It's uh, um, uh, mostly used and most popular are this nutrient media: tryptic soy broth, MRVP broth, phenol rate, carbohydrate broth, growth of bacteria in liquid media, turbidity seen at the end of the broth. How can you know the bacteria is growing or not? If you check the turbidity in terms of optical density, you can understand whether bacteria is growing or not. So we are learning three types of media as we discuss the types of culture media based on consistency of physical state. One is solid medium, other is semi-solid medium, and liquid medium. So here we already discussed what is solid media, what is the liquid media, and what is the semi-solid media, and their examples and their purpose. Now, now we will discuss the types of culture media. Now we will discuss the types of culture media based on types of culture media based on chemical compositions and applications. This is very important. We need to know the chemical compositions, because not we will see, we will learn the all bacteria not will grow in same compositions. They need different composition, because they have a different requirement, because they have a different property. As for examples, there are 7 routine laboratory media. One is basal media, number 2 is enriched media, number 3 is selective media, number 4 is enrichment media, number 5 is indicator media or differential media, number 6 is transport media, and number 7 is storage medium. There are 7 media routinely used. So we need to learn one by one the compositions of this media and their use and their examples. So what is basal media? This media is simple as it enhances the growth of many microorganisms, and it is routinely used medium in the laboratory. It's having carbon and nitrogen source, and this media allow the growth of non-fastidious bacteria without any enrichment source, and this is used for subculturing, and it is non-selective media. Staphylococcus and Enterobacteriaceae mostly grow in this media. And this is the example of basal media is a nutrient agar, and peptone agar is the example of basal media. So let's repeat what is the basal media. This media is simple as it enhances the growth of many microorganisms, and it is routinely used medium in the laboratory having carbon and nitrogen, and this media allow the growth of non-fastidious bacteria without any enrichment source, used for subculturing, and it is non-selective media. Staphylococcus and Enterobacteriaceae grow in this media. As for examples, basal media is nutrient agar and peptone agar. Number 2 is enrichment media. This media requires the additions of other substance like blood, like egg, or like serum as per requirement of the bacteria. And enriched media allow the growth of devised microorganisms but inhibit the other and fastidious microbes grow as they require nutrients like vitamins, growth promoting substance. So we need to find the difference from previous the basal media and the enriched media. Is enriched media is actually added some substance like blood, like egg, like serum as per the bacterial need. And how can it become enriched? And enriched media allow the growth of device microorganism but inhibit other and fastidious microbes grow as they require nutrients like vitamins and growth promoting substance. So this is actually called enriched media. If you, as per example of enriched media, you can say blood agar is the enrichment media, chocolate agar is the enrichment media, LSS, Monsors, Todocolate, Launstein Jensen media. Blood agar identifies why blood agar is used in enrichment media. Blood agar identifies hemolytic bacteria, chocolate media for N gonorrhea, as per examples. Now, after basal media and enrichment media, we'll learn the selective media. As by name, we can tell the media show the growth of selective microbes or desired microorganisms and inhibit the growth of unwanted microorganisms. That means selective media means they will select some microorganism, not all. By name, we can tell this media show the growth of selective microbes or desired microorganisms and inhibit the growth of unwanted microbes. And this inhibition occurs by adding. How can this inhibition occurs? You can add bile salt, you can add antibiotics, you can add dyes, you can add pH adjustment, you can this media is agar based, and any media is possible to transform into selective by adding inhibitory agars. So try to find the difference from basal, from enrich, and with selective media. Selective media means you need to add some ingredients, you need to add some things that will inhibit the others. The inhibition occurs by adding. How can this inhibition occurs? And this inhibition occurs by adding bile salt or antibiotic, or you can add dye, or you can change the pH. So then it will be a selective. And the media and media is agar based. You need to add agar because it is solid media, and a media is possible to transform into selective by adding inhibitory agars. So this is the comparison chart where different selective media is one mannitol agar, Salmonella shigella agar, TCBS agar, as per example, and what is what the bacteria can grow like mannitol agar. It has 7% of sodium chloride. Look at that, 7%. How the large amount of salt. So the it has 7% sodium chloride that inhibit the growth of other microbes and promotes the growth of staphylococci, and it has phenol red dye and that produce acid staphylococcus. Use the mannitol for the acid production, and the color of the phenol red change from yellow red to yellow. There is one is pH indicator and another is salt, and this is the selective for staphylococcus aureus. So if you want to grow staphylococcus aureus, and if you don't wants to grow the undesired organism, then you can use mannitol agar because it has 7% sodium chloride and also phenol red. There's a two indicator, so they will only select the Staphylococcus aureus for their growth. Next examples as for Salmonella shigella agar or deoxy chocolate agar. It is used for isolation of Salmonella bacteria that cause typhoid and that cause typhoid, selective for Shigella. As for example, like McConkie agar, it has bile salt that inhibit the growth of gram-positive bacteria, like selective isolation for Enterobacteriaceae. TCBS agar, the light green translucent media containing bile salt, inhibit the growth of unwanted bacteria, and selective for Vibrio cholerae. If you want to grow Vibrio cholerae, then Vibrio cholerae produce acid by fermentation of sucrose, and that acts as an indicator called bromothymol blue, and yellow colonies appears. So here this is the overall examples of selective media. And Laustin-Jensen media, it has been used, it made selective by adding malachite green and stop the unwanted growth of the pathogens. It is a selective for tuberculosis. So there is a bacterial name like Staphylococcus aureus, the bacterial name like Salmonella shigella, Is bacteria name like Vibrio cholerae, bacteria name like tuberculosis. As we all know, these bacteria are having importance for different disease causing, different disease, different causes, and it has role in important role. So how can they will grow, and how can they will grow? We need to use selective media. Why will you choose selective media? Because we don't we will need will required for their growth, but not unwanted other organism growth. So so thank you. Thank you. This is the introductory session. The elements and their role of media for bacterial growth.